Okay, guys, hi, welcome. Today's class is going to be about an introductory to genetics. But before we get into that, let's just do a quick little ASMR activity. Here is some saliva swirling in my mouth. Okay, great, thank you. All right, so let's get started. Before we begin, let's review a couple of key terms. Up here at the top here, we have four key terms. Allele, traits, gene, and homologous chromosomes. These should be familiar from the last chapter on cell division. One of many alternative forms of a gene, we're going to call that an allele. Segments of a DNA that carry hereditary instructions, what do you think that's going to be? That is your gene. A distinguishing quality or characteristic that can be passed from one generation to another, we'll call that traits. And lastly, DNA sequences that have matching genes but may differ in some or all the alleles are going to be homologous chromosomes. Remember, you get one from your mom and one from your dad. So these four terms are going to be really important in terms of understanding the upcoming discussion that we have on genetics. Let's get started with that. All right, so the two big terms for today are going to be phenotype and genotype. They're similar sounding, but there's a pretty good difference between them. So a phenotype is the outward appearance or the expression of a trait. It's how an organism looks, how it behaves, how it might smell, how its biochemistry work. It's actually the physical, the pH in phenotype kind of stands for pH in physical. It's the physical description of a trait. Genotype, on the other hand, is the combination of alleles that a person gets. So a genotype is going to be two letters. We're going to represent it with two letters. A big C or a little c, a big A, a big F, a big F. So genotypes are just two-letter combinations that result or that give rise to the phenotype. So, for example, if the allele is dominant, we're going to use capital letters. For example, curly hair would be a capital C. If the allele is recessive, it uses the same letter, but in a lowercase. So if straight hair happened to be a recessive trait, then we would use a lowercase c because the uppercase C would stand for curly hair. So it's the same letter, but what distinguishes them is their case, uppercase or lowercase. Tricks to remembering the difference between phenotype and genotype. Genotype starts with a G, and it's the genetic code. Phenotype starts with pH, and it's the physical description. So here's a really clear indicator of what's going on here. When I refer to a genotype, that means your letter combination of alleles, and phenotype refers to the actual physical description. All right, how many alleles do you have for every gene? Well, when you get a gene category, you can only get two alleles for every gene. You get one of those alleles from your mom and one for dad. We represent alleles with upper and lowercase letters. The letter is shorthand for an entire sequence, and that's kind of important to remember and refer back to when we did our DNA chapter. Remember, DNA sequences that code for proteins are huge. They can range from 20 to 100,000 nucleotides long. So when you're looking at this H right here, this allele H, that segment, try to remember sometimes that that's an extraordinary amount of information there. It would look something like this. So here's your A, T, C's, G's all in a row, and all of this combination codes for one of those alleles. So let's say this little lowercase h represents the allele for wavy hair. That letter H represents a specific segment on the chromosome right here, this little purple band, and it has a sequence of nucleotides that looks like this. It's a huge sequence. So just try not to forget about that we're going to kind of leave behind the world of the A's, the T's, the C's, and G's, and the world of the very small molecules. And now we're going to start transitioning to more conceptual things when we talk about genetics and inheritance of traits. So I'm just trying to get you to remember that when we're using alleles and shorthand letters to represent an allele, behind it is a huge amount of DNA and concepts that we've already covered. Remember, this entire code is just to produce a protein. If the protein is just slightly different, it can result in a slightly different trait. And an allele is a variation of a protein. So that's how that kind of works. Proteins are going to be very similar, but when they have slight variations, that will be considered a different allele. All right, how many possible two-letter combinations can you make using the following alleles for hair texture? So let's say the order doesn't matter. So we have big H, we can do one combination would be big H, right? So there's your genotype. Another combination might be big H, little h, There's another separate genotype. A third combination might be little h, little h. There's a third combination, and you might think that a fourth combination could be little h, big H. But it turns out that the order doesn't matter, so this one and this one will be the same thing. Big H, little h, and little h, big H is really the same thing, so we don't even have to do this. Typically, you write the capital letter first. So really, there are 3 possible genotypes that you can get for any 2-letter combination. So some examples of genotypes would be big H, big R, little t, little b, big B, little b, that kind of thing. And then again, phenotype examples: curly hair, tall, widow's peak, sickle cell anemia. These are the descriptions that describe the trait that you've inherited. There are 3 possible genotypes, as we just shown. So when we use the letter H as our example, when it was 2 uppercase H's, we call this homozygous dominant. This means that the genotype has both dominant alleles. Sometimes this is referred to as a purebred, and this will result in a dominant phenotype. A homozygous recessive genotype is when you inherit 2 lowercase alleles. So this genotype has both recessive alleles, and that is also considered purebred, although these terms are kind of getting outdated. This will result in a recessive phenotype. Heterozygous is when you get one of each, a dominant allele and a recessive allele, uppercase and lowercase. The genotype has a dominant and recessive allele. This is considered a hybrid, and it will result in the dominant phenotype just like the homozygous dominant. And that's kind of what is at the difference, or at the heart of the difference between dominant and recessive traits. You only need one dominant allele in order for that trait to come through. But we'll get to that in just a second. So let's talk about that. Dominant versus recessive traits. This explains the relationship between alleles of a single gene. Every organism has certain characteristics called traits. Each trait is represented by that 2-letter code that we just talked about, the big G, the little g. Here we have T's and B's being used. Some alleles are considered quote-unquote stronger, although that's not the best word to use, than others. We call those traits dominant, while others are weaker and they're recessive. But really, the weaker ones just get masked or covered up by the dominant traits. So, some examples of dominant traits. This is a trait that requires only one of the alleles to be present in order to show itself. A genotype combination be homozygous dominant, capital letter, or heterozygous, capital letter, lowercase letter. So here are an example of a couple of dominant traits: brown eyes, curly hair, widow's peak, unattached earlobes. We'll talk about what those are in a second. Dark hair and straight thumb. So here is an example of a straight thumb, and this is a hitchhiker's thumb over here. So some of you may have hitchhiker's thumb, some of you may have straight thumbs. A regular thumb, in order to have a regular thumb, which is the dominant phenotype, you only need to inherit one of the capital letters. So you could have capital letter, or you could have just one capital letter and one lowercase letter. In either event, you'll have a regular thumb. In order to get a hitchhiker's thumb, you need to inherit both recessive alleles, the little t combination, homozygous recessive. If a person's genotype is homozygous dominant, it doesn't mean that they have a more aggressive trait. So it's not like if you have big T, you're going to have a straighter thumb than a big T, little t. All you need is just one big T in order for the straight thumb trait to show itself. Recessive trait. This is any trait that requires both of the same allele to be inherited. So again, with our hitchhiker's thumb, the only way you can get this phenotype is if you inherit 2 recessive alleles. So you need both alleles for this gene category to be recessive in order to have a hitchhiker's thumb. Other examples of recessive traits would include blue eyes, red hair, straight hair, baldness, attached earlobes, and hitchhiker's thumb. A recessive gene is usually the result of a loss of functionality. The DNA sequence for blue eyes means that it just doesn't produce the brown pigment. So whatever protein would normally be created, it just means that protein might be broken. And while that's not going to hurt you, because blue eyes doesn't hurt you any more than brown eyes, or a hitchhiker's thumb doesn't hurt you any more than a regular thumb, it just means that the protein that would normally be made just isn't working correctly, and as a result, you have a recessive phenotype. Now, sometimes recessive phenotypes can lead to bad disorders, but we'll talk about recessive disorders on another class. As an analogy for dominant versus recessive, let's pretend you're looking at a piece of glass. Now, a piece of glass has 2 sides, an interior side and an exterior side. but you only need to paint one side in order for it to be considered colored. You can paint both, but that won't make a difference because once you've painted one side, it's already painted. In order for the glass to be transparent, both sides have to be clear. So that's our recessive situation. Recessive traits work the same way. There needs to be a loss of functionality in both alleles in order for the gene to be considered recessive. With our window pane, as long as one side is painted, that would be our dominant phenotype. It doesn't matter if both sides are painted. As long as one is, then it will be fine. So how would you describe the below chromosomes that have the same genes but different alleles? This is a term from the previous chapter on cell division. Hopefully you would have guessed homologous chromosomes. So up here we have I've tried to make my best model of a chromosome. So here's your mom's DNA. This is one of her chromosomes and another one of her homologous chromosomes. This one came from her grandmother. This one came from her grandfather. So notice that if we look at a couple of these allele pairs or these genes, these two are identical, so they'd be the same thing. They'd be homozygous. Here we have an allele pair we have one gene that's being shown as having vertical stripes and one that's shown as horizontal stripes. Those are going to be different. That's a heterozygous combination. Here's a homozygous combination and again a heterozygous combination because this gene model is different from that one. So let's just focus on these horizontal and vertical stripes. Let's say that the vertical stripes are dominant and the horizontal stripes are recessive. So what that means is that this is going to code for a big B and this one will be a little b. So going down to your father's chromosomes, he has two homozygous copies. So in both cases they're vertical, so his combination, his genotype is big B, little b. During meiosis, these alleles are going to separate during anaphase 1 and anaphase 2. And in the gametes that are produced, they'll contain only one copy of the allele, right? You only want one copy going into each gamete. So the big B is going to go up into this gamete, and the little b is going to go into that gamete. Down here, it doesn't matter because the only allele that the dad can give is big B, so all the gametes will have a big B inside of them. During meiosis, your mom's and dad's genes are separated into haploid gametes. 50% of the gametes get one allele, 50% of the gametes get the other allele. When the sex cells combine during fertilization, they combine to give their alleles, and that gives you a genotype. So in this case, we could have any combination from your mom's gametes and your dad's gametes. So big B or big B little b. So what we use to show combinations is something called a Punnett square. Punnett squares are used to represent the possible, and I want to stress that term, the possible allele combinations in children that are created when gametes combine. So here are your dad's alleles and here are your mom's alleles. That's what goes on the outside of the box. So let's take a look what happens. We can take this B and we'll put it at the top there, and we'll take this little b and put it there, because those are the only two alleles that your mom can pass down. Your dad can pass down a big B and a big B, so we're going to put one of those here and the other one down there. So now, using this Punnett square, we can try to predict what's going to happen or what the possible offspring might inherit. So in this box, the allele combination would be a big B from mom and a big B from dad. In this box, it would be a big B from dad and a little b from mom. Over here, we would have the big B from mom again and the big B from dad. And then over here, something similar. Big B from dad, little b from mom. So here we can see our four possible combinations that can result from these allele gamete combinations. Here we have homozygous dominant, heterozygous, heterozygous, and homozygous dominant. In all cases, every single one of these children is inheriting a big B somehow. So that means that there's no possibility for them to have blonde hair, only brown hair. Oh, I'm going to sneeze. Here we go. Excuse me. All right, let's keep going. What if dad was heterozygous? So instead of being big B, little b, let's say that he was big B. Instead of being big B, let's say that he was big B, little b, which is heterozygous. So in this combination, we have a homozygous dominant. That's going to be definitely brown hair. We have a heterozygous allele combination. That's brown hair as well. Down here we have big B, little b. Again, that's heterozygous, and the dominant capital B is going to win out, so it's brown hair. But in our last category, we have a little b from dad and a little b from mom. And so what that means is that even though the parents both had brown hair, it is possible for them to have a blonde-haired child because they both carry this recessive allele. So that's how you can have different phenotypes from in the children than in the mothers and the fathers. Punnett squares are kind of fun because they can be used as logic puzzles too. So a person you know has a hitchhiker's thumb. This is a recessive phenotype, so that's going to be little t. There's your person. That person's mother also has a hitchhiker's thumb, which we know is recessive, so the mother must also be little t. However, his sister has a straight thumb. What is the genotype and phenotype of the father? So the sister has a straight thumb. We don't know if she's homozygous dominant, big T, or heterozygous. We just know that her phenotype has a straight thumb, and we don't know what the father is. So let's try to solve this. We know that the mother is little t, so let's put one of her little t's here and one of her little t's there. Now because of that, and we know that the person is little t, that means that they got this little t from mom, but they must have gotten another little t from somewhere, which means they got it from their dad. So we now know that at least one of the alleles of the father is little t. Well, now that we know this one and this one, we can figure out number 4. That's going to be homozygous recessive. And we now know that the sister, remember, she has a straight thumb, so she did get a big T from her dad. So that means that we know that the father's other allele must be that big T, which is the allele for a straight thumb. And now with this one, we can solve the other two as being heterozygous. So yeah, that's how you can use Punnett squares to solve for problems. Once you have a Punnett square solved out, you want to be able to express the information in a genotypic and phenotypic ratio. So here is this is the puzzle from the previous page. The genotypic ratio is the simplest ratio of all genotypes in the possible offspring. So here in the offspring we have 1 heterozygous to 1 homozygous recessive. It's really 2 to 2, but you can reduce that down to 1 to 1. Phenotypic ratio is the simplest ratio of all phenotypes in the possible offspring. So we have 1 straight thumb to 1 hitchhiker thumb. It's 50-50%. What's the genotypic and phenotypic ratio of this outcome? Well, we have 1 homozygous dominant, we have 1 homozygous recessive, and we have 2 heterozygous. So our genotypic ratio would be 1 to 2 to 1. Our phenotypic ratio is not going to be 1 to 2 to 1 though, because homozygous dominant and heterozygous are going to code for the same phenotype. So we actually have 3 of these boxes that are going to code for a straight thumb, and only that one box is going to code for a hitchhiker's thumb. Okay, so that's it from me. I hope that serves as a pretty good introduction to genotypes and phenotypes and genetics and Punnett squares. Okay, and if you have any questions, email me or let me know. Thanks. Bye.